Yeah, I'm really excited to be here today. Thanks a lot again. My talk is going to be a bit different on patterning and implants, but that was super interesting to hear the previous talk and the discussion. Um, so I want to start with a fundamental question in biology, or I find it at least fundamental, which is how a bunch of cells that have the same genetic material end up forming patterns with different cell types mixed together that can have different cell sizes, right? So how this happens, because this happens across different tissues. Your ear has different cell types with different shapes and sizes, right? But actually today I'm going to talk about plants, right? So in plants, if you look at the leaves, you're going to see these trichomes, these structures. Do you see my cursor, right? So these are trichomes, and these are huge cells that are scattered throughout the leaf. And if you look on the other side with a confocal microscope, you're going to see these oval-shaped structures that are called stomata. So if I zoom in, you see a bit better the stomata. The stomata are related to gas exchange. And then there's also pavement cells, which would be the rest of the cells mainly. And these cells have a kind of a jigsaw puzzle piece shape, right? And this pave the rest of the tissue. So the question that we have is, okay, how do you get these patterns of different cell types organized in such a way, right? And we will talk more in detail about the pavement cells. But before getting into this, it's important that we mention reaction diffusion models that has been proposed by Alan Turing in 1952. And basically, there was a revolutionary idea that diffusion of chemicals, of proteins, of these biochemical signals would drive pattern formation. So if you have your activator or inhibitor that diffuse with certain conditions and certain nonlinearities, the diffusion would drive these kind of patterns, right? So this framework has been very important for understanding developmental processes, but it's still challenging to link it to actual processes. In the case of cellular patterning, there's been some connection. So for the stomata patterning and the trichome patterning, it's been proposed that there's inhibitors that would orchestrate the patterning process and be important in the spacing of the stomata and trichomes. But actually, what's happening for pavement cell patterning? So there's not much known about the pattern formation if there's any pattern there, right? So to talk about this, let me discuss first what's happening in the sepal, which is a smaller and simple system. So this started for me in 2015 or 14 when I was doing a postdoc, and I met Adrian Rother that was visiting the Sainsbury lab where I was at the time in Cambridge. And she showed us that the sepal in Arabidopsis has these amazingly large cells, and you see them here colored in red, and these cells have a very big nucleus. Okay, so these cells actually endereplicate after doing the synthesis of their genome, and they have 4 copies of chromosomes. They get trapped in an endocycle, and then they become 8C, 16C, 32C. I mean, you name it. They become huge with a very large nucleus, right? So the question was, yeah, how this happens, right? So why these cells? This I did not say, but these giant cells would be a subtype of pavement cells. They look a bit different than the pavement cells in the leaf, but they are still a subtype of pavement cells, right? So how this would happen? And then a collaboration started between the Johnson lab, the Locke lab, the Sainsbury lab, and the Rother lab. And well, first, the Rother lab found that ATM01, which is a transcription factor that is specified that is important for epidermal specification in the sepal. So if you modulate the quantity of ATM01, this will modulate also gradually the amount of giant cells in the sepal, and this has an importance also for the shape of this organ. Okay, so then the next question was, okay, how ATM01 then orchestrates giant cell patterning at the single cell level? And then Heather Mayer, a PhD in Rother's lab at that time, looked at ATM01 fused with MC3 influorescent reporter, what was happening in the developing sepals under the microscope. And José Teles, a postdoc at the Sciences Lab at that time, did 4D image quantification. And then we proposed from this analysis that actually there were at the single cell level, if you look at the concentrations of ATM01, you would see that when you go above a certain threshold, when it happens at a certain time of the cell cycle, the cells would endoreplicate, okay? Whereas if you don't pass this threshold, or if you pass it at the wrong time, then the cells would divide and would not endoreplicate. So then we proposed a model in which there's a very simple network. ATM01 would induce the LIGO, which is a cyclin-dependent kinase inhibitor, and then LIGO, if that was above a threshold in G2, that would prevent cell division and cells would endoreduplicate. Then very importantly, there would be stochasticity because there's intrinsic stochasticity in the reactions within the cells. And then also we had to consider growth effects. And overall, that would be a movie that would show you the multicellular simulation. So you see the blinking of colors is the ATM01 fluctuation at the single cell level that eventually drives this ploidy pattern at the tissue level. Okay, so that was a publication in 2017, and that in a nutshell says that fluctuations at the single cell level, so it's a cell autonomous process, drives this giant cell specification. But obviously, this raised many other questions about how this process was happening, right? And that's what we are doing in our lab, among other things. But the first question that we had is, are there giant cells in other tissues? And how are giant cells spatially organized? Because if we understand their spatial organization, perhaps we can better understand how the pattern is formed and whether this is really a cell autonomous process, right? So is it really a cell autonomous process? So that was a collaboration again with the Rother lab, and the main players of this were Francis Clark and Chihang Wang at the Rother lab. So Francis Clark was a PhD student in Rother's lab at that time, and she was doing the experiments, the imaging, and part of the quantification. And Chihang, an intern, was doing also quantification. And Gauthier Weisbart in my lab, he was a former PhD student, my first PhD student. He did the computational analysis and the theory and simulations. So the first thing are there are giant cells in other tissues. Yes, so that would be a typical giant cell in the sepal. If you look at the colline leaves, that would be these leaves that would be more at the stem, let's say in the stem of Arabidopsis. You would see also these very large, a bit more loby cells. And if you look at the rosette leaves, which are these leaves more at the base, actually you see very big pavement cells that would be giant cells, right? So we wanted to do quantification of that. So we did live imaging, imaging of these leaves in live tissues, and then quantification. So, and we saw that in the sepal, the abaxial side of the sepal, which is the bottom side of the sepal, when the flower is open, there would be indeed very big cells. So here the color code is the area, whereas in the adaxial side, so the other side of the sepal, we would just have small cells. And in the two sides of the leaf, abaxial and adaxial, actually, we could have a salt and pepper pattern of, let's say, of sizes, or we would have a range of sizes of the pavement cells. So we would have giant cells, but we can see this better in the quantification where we are plotting cell area. You see that the sepal has a very long-tailed distribution of areas. And this also happens in the leaf. And you get more or less the same range of sizes. And we know that cell size is related to ploidy, and we get also the same kind of ploidy levels in these systems. So then we looked at the genotypes affecting the giant cell pathway in the sepals and how we look this in the leaves. So that would be the wild type. Then here we can see the ATM01 mutant and LIGO mutants. The LIGO mutant would not have giant cells. The ATM01 would have just a few, 0.7% of giant cells, so basically no giant cells. And then if we overexpress ATM01 and LIGO, we would see a lot of giant cells. And we saw that these trends actually were also happening in leaves. So that's a portion of the mature leaf. Wild type in the ATM01 mutant, we would just have very few or no giant cells. In the LIGO, no giant cells. And then in the ATM01 overexpressed and LIGO overexpressed, we would have a lot of giant cells, right? So we wanted to then give quantities. We wanted to have a definition of giant cells quantitatively. So then, before that, let me just conclude that giant cell specification, we see that patterns the sepals, but also the leaves. And then if we want to get numbers about giant cells, we can look at the wild type versus the ATM01. Then we can establish a threshold of cell sizes in the ATM01 mutant, about which there's basically no giant cells. That was more convenient than LIGO because otherwise we would get too many giant cells in the wild type. And then expert eye said that that was not happening. So then that's what we did. This is a sepal results. So here on the left, you're seeing an area quantification, and color-coded, you see in magenta the giant cells. In blue, we were quantifying stomata through doing other kinds of cell classification analysis. But yeah, now we can have a quantitative definition, a first quantitative definition of what is a giant cell. And we did the same in the leaf, okay? And now then we can start thinking about the spatial arrangements of these cell types, right? Because if you remember, I said that we proposed that there's stochasticity and HMO1 that would drive this pattern. So if that's a cell autonomous process, I would expect that there would be some randomness here. So is there really a random pattern here? So if you think about quantification of random patterns, people use point clouds, like for instance in stomata or trichomes. So the left picture would be an ordered pattern, the middle one would be random, and the right one would be an aggregated pattern. But actually, giant cells size is comparable to the spacing between them, and they have very complicated morphologies in the leaves, and cell size, therefore, cell shape and cell connectivity matters for doing any assessment about randomness in the pattern. So yeah, we cannot use this approach. And Gautier found two papers of the Genovese lab in Paris in the in École Normale. And in these papers, they proposed a computational method in which they can randomize computationally the arrangements of cells preserving their area, okay? So that's a very interesting and complicated method that I don't have time to explain now, but it worked for us. So if you look on the left picture, that would be a segmentation image, and I'm putting numbers, as that you see that the same cells are displaced or rotated in this picture, preserving more or less their shape. So that's what we did in the leaf. That's an image of the segmentation and classification. And then we performed 400 randomizations from each replicate. And you see that the cells are randomized here. And then we had to do some statistics, right? So we compute a certain spatial observable about the pattern, and then we would create a null distribution from our randomizations. And then we have our experimental observation, and based on where we are hitting or not hitting the null distribution, we can also compute a p-value. So if the p-value is low, we are rejecting the null hypothesis of having a random pattern. So the observable we chose was related to giant cell clustering, and in particular, the average giant cell neighbors per giant cell that we would observe. So in the case of the leaf, we observe indeed that giant cells tend to show more clustering than in the randomized leaf tissue. That was very striking here. And we also saw this, we also did the same in the sepal. In the sepal, in the randomization, we had to fix a bit the orientation of this randomization. We also got a very low p-value. So then we could also state that we observe more clustering than in the randomized sepal tissue in terms of giant cells. That was very nice, but if you think about what I told you about the theory, when you have reaction diffusion-based mechanisms, you expect a relatively ordered pattern. However, I said to you that there was a cell autonomous mechanism with stochasticity, so I was expecting a random pattern. So I thought that we had to go back to our model, the stochastic model. So that's what we did. We look at the model and then we treat it as we treated the images. We classified giant cells based on the image, and then we performed randomizations. And then by looking at these tests, we could see that there was clustering also in our cell autonomous stochastic model, which was very interesting. But then how cells would get clustered? Then if you think about divisions, you can think about, let's put this following cartoon. Divisions could be generating this clustering effect. So in an early stage, these magenta boxes would be two giant cells next to each other. So in an early stage, there's less cells, so it's more likely to find neighboring giant cells by chance touching each other. Whereas at the later stage in development, there's divisions, although you have the same giant cells more or less touching each other, then it's less likely to find neighboring cells being giant if you do this randomization algorithm. So that's what we did. So we did this in the computational model. We did this randomization early and late in development from our simulations. And what we observed is that this null distribution would be shifting to the left with respect to our observation in this model. So then what is happening is that at the beginning, the pattern is spatially more random, and at the end is less random. It shows this clustering effect. And we thought that it was also important to show this from data. So we took the paper by the groups of Olivier Hamand and Ernest Wood. There was also Adrian in this paper. And then we classified the images, and there was tracking. That was nice. But then we did our computational randomization. And then we saw also that this null distribution was shifting to the right, sorry, to the left. Meaning that there's this clustering effect, and then there's this decreasing randomness of the pattern because of the cell divisions. Okay, so in a nutshell, what we are learning is that this cell autonomous mechanism, because you have proliferation, you can also get clustering effects in your pattern. So in summary, what I'm telling you is that we learned that fluctuations at the single cell level in HMO1 drive giant cell specification. We also found that the common pathway patterns giant cells in both leaves and sepals. The giant cells exhibit a cluster pattern in the mature sepal and the leaf, and that a cell autonomous stochastic mechanism plus cell divisions might lead to a cluster pattern. And if I may, then I would say that an important take-home message is that aside from cell-to-cell signaling and diffusion-driven mechanisms, cell proliferation can also shape a cellular pattern, right? And if I have a couple of minutes left, then I would like to say that we just published a new methods paper that we try to characterize spatial patterns of ploidy, because patterns of ploidy are ubiquitous across organisms. So in the case of plants, I showed you the sepal and the mature leaf. There's also the root. But in the case of animals, for instance, in your heart is polyploid when you age. Your liver as well. So there's a lot of polyploidy effects, right? So we need ways of being able to quantify ploidy in a non-destructive manner. And because it's known that ploidy scales with nuclear size, we thought that from images, we could do that in a high throughput manner, right? So that was an effort from this Polyploidy Institute that I'm part of. And here you have the website. If you're interested, please contact us. So in this institute, we care about how ploidy affects cells, tissues, organisms, populations, so across scales, and how ploidy also affects these different levels across species. Okay, but back to the methods project that was led by Nicholas Russell in collaboration with the Rother Lab and Fox Lab. And then what he proposed in a nutshell is that you can have your confocal image in 3D or in 2D of your set of your nuclei. And it's optimal that you use a dye or a reporter that in a way relates to DNA content. Then you can do your segmentation in 2D or 3D, ideally in 3D. And then you can do some unsupervised classification, and in particular Gaussian mixing that finally will give you in a high throughput way your spatial ploidy without destroying your samples, right? So basically, you can do this with one nuclear feature. You can use just nuclear volume, and then you're going to get your ploidy peaks. You can use also histone nuclear fluorescence because histones are related to ploidy. But obviously, if you can combine more than one feature, you can get better results that you can compare with your ground truths. In our case, the ground truth was flow cytometry, so using two features was a better fit. And voila, you get your spatial pattern, right? So we hope that people can try this in their own systems. Oh, I forgot to say that in the methods paper we have this tried in two other systems. So in the Drosophila hindgut, if upon injury, there's also the polyploidy, there's also ploidy pattern, and also in the heart of humans, we also saw that our pipeline shows the endopolyploidy pattern, right? So that was work with the Don Fox lab. So please try it in your systems if you are interested, because it would be very interesting to see how these ploidy patterns can be important in development, in aging, and in disease. And that's the same slide. So I already thanked people throughout the slides. And yeah, thanks for your attention.
Thank you so much, Paul. Again, if people have questions, please put them in the chat. Maybe I'll start, which is kind of like, from Animal Land as always. I have questions about why. I mean, so I was really intrigued by the kind of the A that these like sheerly looking at patterns without like it has in a way it is a limited view into what generates those patterns, right? Like you can basically have multiple ways of multiple dynamic pathways to generate the same spatial patterns, and it's not enough to kind of infer, via randomization what's cell autonomous versus not cell autonomous. It sort of can't entirely tell you the cell mechanisms just by the patterns alone. Um, and I'm wondering if you have more sort of thoughts about, well, like how is there like impossible to essentially just look at patterns and, from a static image and tell, do we necessarily need dynamic information at the single cell level to understand these sort of tissue level mechanisms? This is really a large question.
Yeah, no, that's a great question. Well, first of all, that's true that we cannot rule out the effect of cell-cell effects in our system. Actually, there's ACR4 DEC1 that could have cell-to-cell effects, but so we think that our model is parsimonious in the sense that can create
Mhm.
patterns. There could be also contributions in cell-to-cell effects, of course.
Yeah.
Then your second part of the question, whether looking at static image can we infer something about the mechanisms without the need of dynamics? Well, I think that dynamics helps for sure, but yeah, I would say that dynamics would be really, I mean, it really helps because you can have the same static image at the end, and there could be different mechanisms that drive the same pattern, right? At least molecularly for sure. And from the modeling perspective, sometimes yes, right? So it's non-trivial. I would say that dynamical information can make a difference sometimes to distinguish what's the underlying pattern formation mechanism, right? So I would say let's advocate for dynamics if we can.
Mhm.
If not,
Yeah.
then we can try to infer something.
Okay. Thank you. We have a question from Kiki, and I can read it if you don't want to read it. Okay, I'll read it.
Yeah, so
So
that's
when you
what I
say,
meant.
okay, so when you say that there are giant cells in other tissues, do those other giant cells also have unusually high numbers of copies of the genome? Why do you not define giant cells by their ploidy, presumably referring to the methods? And what is the likely functional purpose or evolutionary reason for these giant cells? Does the plant experience functional problems when you artificially cause them to be too few or too many giant cells? A lot of questions.
Yeah, okay, let's start with the first one. So, so there are giant cells. Yeah, there's so first of all, not all polyploid cells that you can see in the epidermis of the leaf, I would not define them as giant cells. Like for instance, in the boundary of the leaf or on top of the midrif, there would be also polyploid cells with the same numbers of ploidy that I was talking about in the sepal, presumably. Well, I have not quantified this, but probably it is the same higher levels of ploidy. But these cells, we don't define giant cells. So the giant cells would be more related to those cells that are polyploid and are related to the ATMO1 pathway. Okay, so you can have other polyploid uh, big cells in other tissues that are non-ATMO1 related, and then I would not refer them to as giant cells.
So
Then
all giant cells are polyploid, but not all polyploid cells are giant.
yes, trichomes, for instance, are also polyploid and have a different morphology. But I was also referring now to the boundary cells in the leaf that I would not call them giant cells, but they are big. But we call giant cells more these scattered cells that you see throughout the blade, okay? And that are responsive I mean, that are related to the ATMO1 pathway. Um, why you don't, I mean, the other question is why you don't define giant cells by ploidy? Yeah, so because of this reason, right? Now we are working on more about this concept of how we define giant cells based on nuclear information, actually, but that will be another talk that I would need more time. Then, what is the likely functional purpose of or evolutionary reason for giant cells? Well, we don't work on function, but Adrian Rudder, that is perhaps in the audience and can say also about this, giant cells, if you perturb the amount of giant cells, they found that it affects the morphology of the organ, so the sepals would be looking different, and also the leaves would be having a different curvature. So it affects curvature, right? And the Rudder lab has also shown that it would be so if you mutate if you in the LIGO mutant that is related to giant cells, there would be also effects in immunity, right? So presumably there's it's related to immunity. So yeah, there's potential important functions that we are seeing from giant cells, but this will need more studies in the future. Evolutionary reason for giant cells, that's a super interesting question that I don't know how to reply now, but yeah, polyploidy often is related to stress, and so yeah, that's a very interesting question to look in the future. Um, yeah, so that's basically it. Trying to reply shortly the question. Should we go for the next question or?
Yes, sorry. I think Matthew had a very similar question, so maybe
Yeah.
she wants to elaborate.
Well, yeah, I think you kind of dealt with the first part of what I was going to ask. I guess the second thing is, if you think about the role these giant cells have in shaping the curvature of a tissue, is there clustering contributing to that? Does it matter that they're in the same place? Will that change, the kind of the structure of the tissue in a way that if they weren't all clustered, you would then have different tissue structures?
That's a very interesting point. So I need to think if we have any phenotype that does not show this clustering, but has the same fraction of giant cells. So that would be very interesting to see whether the clustering has an effect in the function and not the fraction of cells. So I don't know, but that's something to look into in the future.
Yeah, and
Thanks.
presumably, not that I could ever do anything like this, but presumably you could, physically model how the different sizes play a role in shaping the curve.
Yeah.
Understand
Um,
theoretically whether there's a possible contribution.
Yeah, that's potentially another contribution. Yeah. Let me also say something on top of your question about the clustering, but that's just a speculation.
Hmm.
You can think about the network of cells, the cells as a network, right? So you can compute, for instance, observables of how easy it is to go from one cell in one part of the leaf to the other cell, right? So these hubs of clusters of giant cells could have potentially influence in transport, for instance, or in the way cells from one part of the leaf are connected to the others. But that would be just speculation, right? But these clusters could have an
Yeah,
effect in this connectivity. Um,
cool. Yeah, that's super
Um,
interesting. Thank you very much.
Thank
Welcome.
you. I have one more question for you, Paul.
Yes,
Which is
please.
like so your comment about the sort of these spatial structures possibly contributing to the gross anatomy of these organs made me think, if you look at other sort of related species, is there a way we can get at some of the more, I guess, tissue level mechanic stuff by looking at, whether, I guess there might be other flowers that curved in different ways or they have different I don't know, patterns of or structures to the organ, and whether the distribution of the giant cells can give you some information about that, or if there's some, I guess, is there a way to like sort of biophysically characterize whether the mechanical properties of these giant cells scale differently from the diploid cells?
That's a very interesting question. I mean, I have to say that I mean Adrian also can say something about this. There's presumably giant cells in other plants, but uh, we have not looked at what's the mechanical implication across these different species. But certainly it's something that I think that it will be important. I have to say that there's other groups that are working more on the mechanical aspects. We have not
Mm.
worked on mechanical aspects, but definitely it's a very interesting question. Yeah.
Okay. Thank you so much, Paul. And thank you again, Yetta. Oh,
Thanks to everybody.
actually, we have one more if you want to take this. We have one more question from Ramiro.
Okay.
Um,
Hi. I was really nice talk. Thanks a lot. I was wondering if you have evaluated, particularly in leaves, if you have evaluated that there are changes in the number of giant cells of the ML1 pathway related to ambient conditions. So, for example, to leave space for more stomata under water stress conditions or whatever. And this also taking into account that other that genes related to LGO are induced under stress, like all those CMS related genes and that stuff.
we have not done this, so in my group. So I don't know if the Adrian's group has done that, but at least it's not published as I recall. I guess that that's something that could happen. I mean, throughout development, also the first flowers would have less giant cells, and then flowers that happen afterwards they would have so that the number of giant cells changes throughout development in the same plant, right? So I would expect that this might be something that can be tuned. So yeah, it will be very interesting to see how environment can tune that, because this can have also an effect in the curvature, right? So I think that there's a lot of things to do in the future about how environment shapes this pattern. Yes.
Okay, thanks. And just another short one. You mentioned that the cells at the beginning of the question time. So you mentioned that in the margin of the leaves, there are also very big cells.
Yeah.
So the pathway to generate those cells is different as the ML1 for the epidermis, bulk epidermis.
I mean, we have not worked on those cells, so I cannot tell you. We have not actively looked at that. But yes, I would say that it would be a different pathway. I mean, we
Okay,
have data, and Adrian has data that could comment on that more. But yeah,
okay, great. Many thanks.
welcome. Thanks.
All right. Well, with that, thank you again to Paul and Yetta and everyone else for coming, and hopefully see everyone in 2 weeks. Okay.
Thanks. Thanks again.
Thank you.
Thanks, everyone.